Hello, I'm Vladimir Brusic, and today I will be talking about big data challenges on a bioinformatics topic, which is single-cell transcriptomics. This is me, and I'm currently in quarantine in China for 14 days. That's why I couldn't participate via video, and masks are now daily life for us. My presentation has 5 parts, and I will start with explaining some background. The central dogma of molecular biology tells us that genetic material, genes in our cells that are encoded by DNA, are transcribed into RNA, and the RNA is executable code for translation into protein, production of proteins. We have big data tools, which we call OMICS, genomics, transcriptomics, and proteomics. And today we will be focusing on transcriptomics. This is the technology that I will be discussing today. We can see that the numbers are huge, 3.2 billion base pairs of DNA. We use 100,000 probes approximately to study transcriptome in our cells, and these transcripts, they actually produce much more than 10 to the 6 of peptides. On this figure, we see how biologists represent central dogma. What is more important is that we have many applications of transcriptomics. I will focus on clinical applications, and we use it to define what healthy means. We want to understand what causes the disease, how to diagnose and perform prognosis, how to select optimal therapy and monitor the course of disease, or assess the effects of particular treatment. How do we define transcriptomics? Simply, it is the study of the transcriptome, which is a complete set of RNA transcripts, either in a single cell or a tissue that we are studying. And this transcriptome is produced by the cells using transcription from DNA, and we study it to see different circumstances like healthy versus disease, or different developmental stages, and so on. Single-cell RNA-seq, or single-cell measurement of single-cell transcriptome, is shown on this diagram, which starts from the upper left corner and then goes clockwise. And we can see that sample is taken from the tissue, single cells are isolated, then RNA is generated from the sample, and after several steps of creating sequencing libraries, we perform sequencing, and after that we have computational analysis. Usually we do clustering, but my group is working on supervised learning, and one of the tasks may be to identify cell types from the sample. The reason why we perform single-cell transcriptomics is as compared to bulk analysis, where we have multiple cells and we take bulk transcriptome, is that individual cells may have different profiles. And if we do bulk analysis, for example, here we can see that 3 genes will be all present in the sample, but only by using single-cell transcriptome analysis, we can find out what is the actual profile in each individual cell. People who work in transcriptomics, they focus on this great promise that we can do many things, like screen for multiple diseases, understand their causes, estimate the progression of disease, and assessing likely responses to particular treatment that is prescribed. And this can be done all in a single experiment. Let's move to the second part of the talk, and see what kind of data we have and how these data are generated. The technologies for single-cell transcriptomics are about 10 years old. In 2009, Tang and his group he have reported the first single-cell transcriptome, which only had a few genes, a few transcripts identified. But over the last 10 years, the technology was steadily improving, and we can see on the top of the slide, from manual to multiplexing, inclusion of fluidics, then robotics, and finally we have different large-scale technologies for generation of transcripts and measuring them. We can see also that from small number of cells, the number on the graph on the bottom, we can see that it's moving to the upper right end, and we can see those split-seq and 10x genomics in the upper corner. And these technologies can produce more than 100,000 screening of individual cells, and they can be done quite fast within one day. So basically, this field is driven by technology, but the bottleneck, as usual, is our ability to analyze the data. Bibliometric analysis is shown on those two slides, and we are focusing on 10x technology because it's rapidly growing area. Single-cell transcriptomics is about 10 years old, while 10x technology is only 2 years old. Since it has been put in broad use. And we can see that 10x technology shown in red on the left graph is rapidly growing, 10-fold growth every year so far. And on the right side, we can see that the number of studies in single-cell transcriptomics mainly focuses on samples from blood and cancer. And the similar situation is with 10x technology. 10x technology is attractive because we can measure expression on more than 30,000 genes, and in each study we can pull out more than 100,000 cells, and in each of those cells we can capture anywhere between 500 to 1500 of genes or transcripts. And the advantages is that 10x is cost efficient, and it has a very strict protocol that can be reproduced in different sites, and it is also fast. Everything can be done within one day. Disadvantages is that on average we can capture only 1,000 genes per cell. Alternative technologies can capture up to 5,000, but they are expensive, slow, and they have smaller number of cells. So we have excellent potential to apply statistical and machine learning techniques. My group has collected more than 2,000 data sets, 2,000 different files, and they include 10x demonstration data from the company side. We have a lot of data, more than 1,900 from GEO, from NCBI, and we have several other data sets from other collaborators. Interesting thing with this data is that we have rich metadata. This is description of experiments and description of the data that we have in our measurements, and they have actually quite a lot of information about the conditions of experiment, types of cells, about how the samples are collected and combined. And it is very important for future study that we have detailed metadata. This is an example of a matrix, the data that we are getting. And we can see in column A vertically, we have list of genes. And on the vertical, horizontally on the top, we have numbers 1, 2, 3, 4 that designate each cell. Normally, this would be some kind of a tag, a label. And we can see that this is a sparse matrix where there are a lot of zeros, and there is here and there we can see a measurement of transcripts that are appearing. Combining data from different studies, and we have 2,000 data files, is not a trivial exercise. And one of the problems is that those studies don't have the same number of genes which are done because libraries are custom made. And we have multiple versions of the genomes, so vocabularies are different. And so we have spent about 2 person years just to clean this data, and now we have a data set. In humans, we have nearly 1,000 files, and we can now do comparative analysis between different studies. The study that we are talking here about is focusing on blood, and we are focusing on healthy human blood so that we can define what healthy means. And we accumulated about 400 data sets. In each of those sets, we have between 500 and 12,000 cells, and we have exactly 30,698 genes and measurements for them. 200 represent healthy PBMCs, and about 100 of them are pathology data sets. And we can see here that we have about 2 million single cells in this data set of healthy samples, and this produces about 6 times 10 to the 10th of data points. And PBMCs have 5 major cell types, more than 100 defined subtypes, and we have about 30 defined profiles in there. What is important is that we need to understand that we have many dimensions of our data set. So different studies have identified that there are many subsets of blood cells. This particular study, one of them, indicate that there is, in their study, they identify about 120 different subsets. And what is important is that we understand that we have 4 cellular dimensions. So hematopoiesis is the program which is embedded in cell development, activation status, maturation state, and tissue or organ of origin of the cell. They all have different transcription profiles. And we also have organism dimension, age, so disease status, development of progression of disease. Also, we can have general status, general health status, and there are also individual differences between people. To better understand these concepts, we are going to discuss a case study. Our samples are collected from PBMCs, which is peripheral blood mononuclear cells, and they're used for standard laboratory testing. And the first part of the procedure is very standard. So peripheral blood is put in the tube, and separation medium is added, and then it is centrifuged for a certain amount of time that is sufficient to separate the plasma, which is no cells, and PBMCs and granulocytes and red blood cells. And from that, PBMCs can be taken and studied further. Coming back to cellular dimensions, this is a program that from progenitor pluripotent stem cell, all different subtypes of blood cells are generated, and it goes through multiple stages, and we can see that erythrocytes, platelets, neutrophils, monocytes, and all other cells are generated in multiple steps. Peripheral blood mononuclear cells, PBMCs, they consist of lymphocytes, T lymphocytes, B lymphocytes, NK cells, natural killer cells, monocytes, and dendritic cells. So there are 5 main subtypes that we are studying. Standard way of getting the subsets of the data is that we use the fluorescence activated cell sorting, which has laser and microcapillary gating, and we use in those experiments, we use antibodies, panels of antibodies that label different subtypes of cells based on their markers, and then through the laser system they get sorted into different bins so that we can study them further. Sorting by fax machine is actually expensive and time consuming, and our idea is that we can actually skip that step and do it all in silico. We can use algorithms, develop new machine learning methods so that we can directly assess what is a particular cell subtype in a sample, and then we can separate it just by separating the files for further study. We have published a paper on classification of 5 cell types using artificial neural networks, and this paper was published just a few months ago, and I will be talking more details about that. In this study, we used 27 different data sets that have B cells, dendritic cells, monocytes, NK cell, and T cells, the major types of cells in PBMCs. And then we were doing incremental testing and training and testing. And the idea is that we can use different data sets, combine them together, and we can create a general prediction system. Our artificial neural network is very big. It has 30,698 input units, which is the number of genes in our list, and 10 header layer nodes and 5 output nodes that correspond to 5 major types of cells inside PBMCs. And the technical details is activation function is ReLU, batch size of 200 for training, and optimization method Adam. Our study involved incremental refinement, and it was done in 7 cycles, and the first 3 cycles comprised twofold cross-validation. We have large amount of data in data sets, so we don't need manifold cross-validation. Twofold is okay. And the next 4 cycles, we did training and testing using independent experimental test sets. Internal cross-validation, in which we take each of the data set is split into 2 approximately similar size parts, and one is used for training. They combined, and one is used for training, another one is used for testing. The results show very clearly that artificial neural network is learning for each specific data set, and the accuracy of classification is about 98% on average. Here we show sensitivity and specificity, which are about 96-97% each. So basically, it means that if we present a data set, artificial neural network will learn it. However, testing with independent data sets, those which were not used for the training and split into 2 parts, but the complete set is independently used for testing, we can see that the results are not as good, and the accuracy after 7 cycles is only close to 90%, which is insufficient for developing blood tests. We attempted to perform feature extraction, reduce the size of the data set, because we have sparse matrices, and for quite a few of genes, there are zeros in all of the data sets, and we started eliminating them based on different influence of particular genes, how many fold they're bigger than others in different sets that represent different subtypes. And we have done about 10 different simulations to see what is happening. We have created a protocol for this analysis, an algorithm, and this was done formally, so it enabled us to do internal cross-validation as well as prospective validation. After 32 steps, individual steps of testing, training and testing, we could see that these graphs show study part which is without feature extraction, and we can see on the right hand side that in different steps we have oscillations. So as we are adding new data sets, accuracy of predictions goes a little bit higher, then a little bit lower, but after about 20-25 steps it starts converging and becomes quite stable. Of all feature extraction simulations, the best one was the second, where we only eliminated transcripts whose values were consistently 0, and it was reduced to about 7,000 to 8,000 features. And we can see that in this case, adding new data sets in cyclical fashion resulted in convergence, so high accuracy has been achieved, but without those oscillations in accuracy in earlier stages, which we can see on the right-hand slide. Our project is at relatively early stages, and we have just demonstrated the proof of principle that we can use supervised learning, specifically artificial neural networks, and we can do classification of major types of cells with relatively high accuracy. We have achieved accuracy at the level of anywhere between 92 and 94%, and this encourages us to do further analysis, development of more models, and the deployment of new data sets. And the practical applications of this may include detection of rare cell types, rare subtypes. Examples are circulating tumor cells. We can use this technology to assess tumor heterogeneity, understand the biological mechanisms, and we can also discover new biomarkers. And the most important for our study is that it encourages us to continue working on design of new blood tests. To bring this project and this technology to the next level, we actually need supercomputers. The analysis of single cells and single cell transcriptomics is a major part of that analysis, is actually considered to be a breakthrough technology, and it is considered to be a technology that will come first to fruition of the currently most promising technologies. So it is a breakthrough technology. Current assessment estimates are that it will take 10 years to come to fruition, and it is going to change medical approach to autoimmune disease, cancer, and I would say infectious disease as well. So we provided a proof of principles that we can accurately classify healthy cells. And the next step is to apply this for discrimination of healthy samples from pathological samples. And we are already working on several diseases with where we want to discriminate types of cells which are involved. And this was done using small data, and it takes a lot of resources, lots of weeks to perform thousands of training and testing cycles. And to actually do proper big data analytics, we need to have supercomputing facilities. And some of the tasks would be to do optimization of the models and then perform comparisons with millions of known profiles. We are now in a situation where we have converging technologies of single cell analysis and supercomputing and artificial intelligence. And we need to put those technologies together so that we can build, optimize, and apply computational models that are necessary for design of new generation of blood tests. And from computer science side, we need ultra-efficient algorithms to be able to screen millions of cells from incoming blood samples and provide medical laboratory grade results. Now we have come to the final part of these talks, the conclusion. What we learned from our study is that artificial neural networks they are actually suitable models for this problem, and the accuracy of classification is up to 95% at the moment. We also tried support vector machines, and they were not as good for solving this problem that involves sparse matrices. And we found that artificial neural networks can be trained without feature selection, without dimensionality reduction, and overall results are very similar. But with the feature extraction, we have smaller, more compact artificial neural networks, still pretty big, 7,000 to 8,000 features. And so this reduces computational load. And when we talk about millions, tens of millions, hundreds of millions cells to analyze in a single study, it can be quite a big overhead. And to cover the solutions place of healthy cells, we need multiple diverse data sets. Probably we need a couple of hundred data sets to cover the range of healthy. And when we talk about disease states, we will have much larger data sets. We can talk about hundreds of thousands of different data sets. So we have assembled, collected, cleaned, and standardized more than 2,000 data files, and they have rich metadata, which is constantly updating. In our case study, which focus on PBMCs or peripheral blood mononuclear cells, the accuracy of cell classification is between 92 and 95%, depending on the subtype of the cell. What we need is 98 to 99% accuracy for clinical applications. So what we need to do is we need to have refined cell ontologies, and they have to be suitable for computational analysis. And this ontology will additionally increase the complexity of our problem to solve. And the overall goal of this project is we want to develop the system that can perform automated analysis of single cells that will be streaming from the machines. And we need supercomputers to define this step because we have 10,000 cells, 100,000 cells, which means that it is highly the problem and the task is highly parallelizable, and I actually would like to invite collaborators on this project, those who have interest. I want to finish today's talk by repeating a great promise of clinical transcriptomics. It gives us opportunity to screen multiple disease, understanding their causes, and considering treatments and the responses to the treatments, all in a single transcriptomics experiment. This is real future, the science fiction, which is actually happening in our real time. I want to acknowledge people who have contributed to this work. There is a group of students from University of Nottingham, Ningbo, China, who have been working very hard on collecting, cleaning data, developing algorithms, packaging. This is quite a big project. And we also have support from Boston University, Computer Science Metropolitan College, from Zhang Guanglan and Lu Chitkushev, who were helping with putting things together and making the system, which is practically useful. And Darren Keskin from Dana-Farber Cancer Institute is helping us with the experimental data.